Hello friends, in this video I'm going to take the detail of tryptophan metabolism, one of the aromatic amino acid. So let's get started. Tryptophan. I told you in the side chain of tryptophan there are two big rings in the side chain, and that is known as indole ring or indole nucleus. And there's no need to learn the exact structure of tryptophan. You just need an idea that there is a big structure, indole ring, which is like two cyclic rings present here in the side chain. So that's why, because it is very big ring which cannot be made in body, so it is essential that we have to take it in diet. It is bulky amino acid. It is a bulky amino acid which is taken in diet, and this bulky side chain which is there in case of tryptophan, this cannot be accommodated in alpha helix. In alpha helix, there is less space there, so tryptophan cannot be accommodated there. So that's why it is never found in alpha helix. And these rings, the side chain, whenever the side chain is rings means non-polarity, so tryptophan is non-polar. So tryptophan is essential non-polar and not found in alpha helix. Now coming to the uses of tryptophan. One use is formation of serotonin and formation of melatonin. Serotonin further forms melatonin. Other use is formation of niacin. Niacin, you know, is vitamin B3. You know, vitamin means a thing which is not formed in body. We have to take vitamins in diet, right? But you can see here that here one amino acid is forming one vitamin. That is, amino acid tryptophan is forming a vitamin which is vitamin B3 or niacin, and this vitamin, because it is getting formed in body, so it is called atypical vitamin. Atypical vitamin, because usually vitamins are not formed in body. We have only two atypical vitamins. One is vitamin B3, other is vitamin D. Only two are atypical. When I'm talking of atypical vitamins, then do not consider those vitamins which are synthesized by intestinal bacterial flora. No, those are synthesized by bacterial enzymes, right? But these two vitamins are synthesized by human enzymes. During our metabolism, we are synthesizing them in our body, right? So only these two are atypical, right? One more use of tryptophan is that it is the donor of formyl group to one carbon pool of the body, and where is this formyl group used? This formyl group is used for giving carbon to the C2 atom of purines. In purines, C2 is contributed by carbon dioxide, and that is coming from formyl group which is donated by tryptophan, right? And one more place where it is used is in the formation of amino acid formyl methionine. It is not in eukaryotes, it is there in prokaryotes. You know, the first amino acid during translation which is added is methionine in eukaryotes, but formyl methionine in prokaryotes. So this formyl group is also taken from formate, which is coming from tryptophan amino acid, right? So now let's take them one by one. First, I will be taking the synthesis of niacin amino acid, which is done by a pathway, a special pathway in body known as kynurenine anthranilate pathway. So let's take this pathway here. Tryptophan is taken by enzyme tryptophan pyrrolase enzyme. This enzyme is very special. It is a heme containing enzyme. Also, it is dioxygenase, that it is incorporating two atoms of molecular oxygen into the substrate, right? Now, the product which is formed is N-formyl kynurenine. N-formyl kynurenine is formed. After that, multiple steps occur here, and 3-hydroxy kynurenine is formed here. This 3-hydroxy kynurenine has two roots. One root is this major one, which I'll be taking, and first it is minor root. In this minor root, this 3-hydroxy kynurenine is forming xanthuranic acid. but this is minor root in the body. On the other side, the major root is forming 3-hydroxy anthranilic acid, 3-hydroxyanthranilic acid, and the enzyme responsible here is very important, kynurenine hydroxylase enzyme, which requires vitamin B6. So if vitamin B6 is not there, that is B6 deficiency, there is excess of 3-hydroxykynurenine in the substrate, which is now taken up more towards the formation of xanthuranic acid. So in B6 deficiency, this xanthuranic acid will be coming in urine of these patients, right? Now let's take this pathway further. After this 3-hydroxyanthranilic acid, many steps occur here and niacin vitamin is formed. No need to do the detail of these steps, but one enzyme which is very important here is QPRTase. Q stands for quinolate phosphoribosyl transferase. Quinolate phosphoribosyl transferase. This enzyme is rate limiting enzyme, very important enzyme, rate limiting enzyme in the formation of this niacin vitamin, right? So this is known as kynurenine anthranilate pathway, and here you have seen that vitamin B6 deficiency will lead to xanthuranic acid coming in urine. And can I also say that deficiency of vitamin B6 will also lead to vitamin B3 deficiency, and that is niacin. Because if B6 is deficient, these steps will not occur. So niacin is not formed in body. So in B6 deficiency, we also are having pellagra-like symptoms. That is, deficiency of B3 is known as pellagra, right? And you know that 60 milligram of tryptophan is taken. 60 milligram of tryptophan is taken in body to make 1 milligram of niacin. This is very important, one-liner MCQ which can be asked. Now next I'm going to take synthesis of serotonin and melatonin from tryptophan. Synthesis of serotonin and melatonin. So here we are starting again from tryptophan. The first enzyme is tryptophan hydroxylase. I told you this is one of the aromatic amino acid hydroxylase, which will require tetrahydrobiopterin also and NADPH also here. And here this is the rate limiting enzyme. Very important enzyme here. So as the enzyme is hydroxylase, we are adding hydroxy group. 5-hydroxytryptophan is formed. We are just adding hydroxy group here, right? Now after hydroxylase enzyme, now the next enzyme is decarboxylase, that is aromatic amino acid decarboxylase. This is a simple decarboxylase occurring here, simple decarboxylation reaction occurring here, which requires vitamin B6. If you remember, oxidative decarboxylation requires vitamin B1, right? But here it is vitamin B6. So after decarboxylase, carbon dioxide is removed, so amine is left, and this is known as 5-hydroxytryptamine. From amino acid removed, so we are left with amine, 5-hydroxytryptamine, which is also known as serotonin. Now further from the serotonin, acetylation will occur. After acetylation, methylation will occur. There are one or two steps here. Methylation will occur. This methyl group will be coming from SAM, acidinosyl-methionine, and what is formed is melatonin, also known as acetyl methyl serotonin. Acetyl methyl serotonin or melatonin, that is formed, and the synthesis of these two compounds are occurring in different cells of the body. First, let's take serotonin. Serotonin is synthesized in intestine and placenta. It is synthesized in the argentaphin cells. It is in argentaphin cells, and this serotonin is a neurotransmitter. It is a neurotransmitter which is responsible for mood elevation, also responsible for GI motility, temperature regulation, and the catabolic or excretory end product of end product of serotonin is 5-hydroxyindole acetic acid. 5-hydroxyindole acetic acid is the excretory end product of serotonin, right? On the other hand, melatonin is also a neurotransmitter which is synthesized in pineal gland, and it is responsible for the biological rhythm of the body. Now let's do the diseases in tryptophan metabolism. One of the disease is Hartnupp's disease. Hartnupp's disease, which is autosomal recessive. There is failure to reabsorb and also absorb neutral amino acids and tryptophan, because there is one common transporter for neutral amino acids and tryptophan. The transporter is present in intestine also and in kidneys also. This transporter is defective here, so this patient is unable to absorb this tryptophan, mainly tryptophan amino acid involved here. This patient is unable to absorb it from intestine and unable to reabsorb it from the kidneys also. So what will be the clinical feature? Clinical feature of these patients, one is amino aciduria, amino acids coming in urine, especially tryptophan coming in urine, and these patients are having pellagra-like symptoms. Why pellagra-like symptoms? Because if tryptophan not available in body, then niacin will not be formed. That's why pellagra-like symptoms will also be there in this patient. And also the tryptophan which is not absorbed, from intestine. Now in intestine, it will be taken up by bacterial action. In intestine, bacterial action occur on this, and indoxyl compounds are formed here. Which is responsible for giving blue color to the diaper of these patients. Blue color diaper, right? And also in intestine, this tryptophan will be forming indican compounds. Indican compounds are detected by Obermeyer test. So this Obermeyer test will be positive in Hartnupp's disease, right? Then coming to the next disease, that is carcinoid syndrome, in which there is tumor of agentaphin cells releasing lots of serotonin. So biochemically, what is happening in the body, that this tryptophan amino acid, too much of tryptophan amino acid is used for the formation of this serotonin. Too much tryptophan used for the formation of serotonin, so that very less tryptophan available for niacin formation. So therefore niacin deficiency will occur in these patients, leading to pellagra-like symptoms in these patients, right? So in clinical features of these patients, there is profuse sweating, flushing because of excess of serotonin here, and GI motility is also affected because serotonin is responsible for GI motility. And in these patients, 5-hydroxyindole acetic acid will be coming in urine, because that is the excretory end product of serotonin, right? This is it with tryptophan. Thanks for watching.